In this section, we will look at the role of ordered water and the distances over which intermolecular binding interactions are felt. And so within this section, we will look at the role of water and the role that this plays in determining how a drug molecule can bind to its intended target. And we'll also look at the distances over which intermolecular interactions are felt and the implications for the strength of each type of binding interaction. Now, biomolecules and drugs exist in an aqueous environment, and as such they are solvated by water molecules. So water molecules are free to interact with both the drug and the binding site in a macromolecular target, and these water molecules can cause problems when we're trying to introduce a drug molecule into the binding site. So these water molecules can interfere with binding interactions between the drug molecule and the protein. Now, removal of water requires a lot of energy. Therefore, the energy that the system gains by adding additional binding interactions between the drug and its target must be higher to overcome this barrier. So here we can see a binding site, and we have our protein and we have water molecules around the side chains of the protein. We also have our drug molecule, and again we have water molecules around it. Now, to enable this drug molecule to bind within the binding site, we have to get rid of all of these water molecules, because these water molecules are in the way of the drug molecule entering and binding. Now, this is a process called desolvation. Now, it takes energy to desolvate functional groups. It takes energy to break the interactions between the water molecules and the drug, and also the water molecules and the protein. So we have to use up energy in order to break these interactions, which means that to get rid of them, we have to lose energy. However, when the drug molecule then binds, it can form its own binding interactions with the protein. This results in an energy gain. And so we've got to make sure that the formation of binding interactions here in the binding site between the drug molecule and the protein gains more energy than we lose by desolvating the binding site and getting those water molecules out of the way. Now, by having non-polar regions in our molecule and in our protein, we can increase the stability of the interaction between the drug and the protein itself. Now, the reason for this is because of the expulsion of water molecules between adjacent regions. Now, if we look at our drug molecule here, this light blue square represents a hydrophobic site within the drug. Now, this dark grey site in the binding site also represents a hydrophobic region. Now, water cannot solvate non-polar or hydrophobic regions, and therefore it will tend to form a more ordered structure around those regions. So it will form a layer where the water molecules are interacting with each other, which is what is represented by these triangles. Now, entropy is decreased due to an increase in order. But when the drug enters into the binding site and the two hydrophobic regions are next to each other, they can form positive binding interactions, and this displaces water molecules. So it will displace the ordered water molecules out into the system. Now, this causes an increase in disorder or an increase in entropy because the water has been displaced. Now, this process actually results in an increase in binding energy, and so it helps our drug molecule to overcome the energy barrier of having to get rid of water molecules by having hydrophobic regions in both the drug molecule and the binding site. And when the drug molecule enters into the binding site, we get this release of water molecules, which results in an increase in entropy. And this is energetically favourable, and it means that this results in an increase in binding energy between the drug molecule and the binding site. So we've now reviewed all of the important binding interactions that a drug molecule could have with its intended target. But something that we now need to consider is how far of a distance these binding interactions are felt, so that we can see which types of binding interactions are important for driving the initial attraction of a drug molecule into its binding site, and which are important for securing the drug molecule in place once it's arrived at the binding site. Now, if we look at the distance dependence for each of these binding interactions, we can see that the distance dependence for electrostatic interactions is 1 over d. Now, for ion dipole interactions, we have 1 over d squared. For dipole-dipole, 1 over d cubed. And for van der Waals interactions, 1 over d to the power of 7. Now, what this means is that an electrostatic interaction is the type of interaction that is felt most keenly over distance. Now, what this means is electrostatic interactions are key in driving the initial attraction between a drug target and its binding site. Now, ion dipole interactions are also felt keenly over distance, albeit not as far, and they can also play an important role in driving that initial attraction. However, dipole-dipole and van der Waals interactions in particular are only felt over a very, very short range. As the molecules get more distant from the interaction sites, the strength of that interaction, especially for van der Waals interaction, drops off exceptionally quickly. And so dipole-dipole and van der Waals interactions are only felt when the molecule is in very close proximity to the groups in which it can have that interaction. And so what this means is that electrostatic and ion dipole interactions are very important for driving the initial attraction of a drug molecule into its binding site. Now, in contrast, dipole-dipole, hydrogen bonding, and van der Waals interactions hold the molecule once it's in position, but they are not felt over a long range. So by knowing this, we can build in a mixture of binding interactions into our drug molecules. So we can mix in electrostatic and ion dipole interactions to drive the initial attraction of the molecule into the binding site. And then we can add in things like dipole-dipole, van der Waals interactions, and hydrogen bonding in order to make sure that the drug molecule is held securely in place. Now, in the next section, we will discuss protein structure. Now, this will include primary structure, secondary, tertiary, and quaternary.